All right, so in the first part of this discussion about monohybrid crosses, we introduced Mendel and the phenomenal discoveries that he made using some simple things, peas, where he was just growing peas and doing some crosses and seeing results. And the results from that look like this, right? He was testing this concept of blending inheritance, and he took these parentals, he crossed them, and he expected in the children, the filial generation, to see some kind of blend, but he didn't get that, so he carried it on further. And what does he see? He sees two really important signature elements. He sees that he takes this trait, color, it's got two phenotypes in this case, green and yellow. The first thing he sees as a signature is that one of those phenotypes, colors, disappears in the F1, right? It reappears in the F2, right? The second thing he sees is that when it does reappear, it's in a fairly standard ratio of 3:1. So that's an important concept for him, okay? So from that, what did he get? Well, the first idea is this. He said, for this to happen, for this 3:1 and this 1 to 2:1 genotypic ratio, that means there's 4 parts, right? How do you get things mating and giving you 4 parts? Is that they must be in pairs. And what he's talking about in pairs are these characters. Now, again, he's thinking in terms of pangenes and those being separate. Nowadays, we know we're talking about chromosomes, but he basically says that the genes must occur in pairs. In other words, you get 2 of them. You get one from one parent and one from another, at least copies of those genes, okay? Today, we would say that the organism is a diploid. In fact, these peas are diploids, just like humans and most mammals and things are diploid. We have 2 copies of every gene, 2 copies of every chromosome that we receive. And we talk about alleles. Okay? Alleles, he called them unit factors, but alleles, that's the idea that you get one piece of genetic information from one parent, one piece from the other parent, and it's the same gene, same thing. We're going to call those alleles, and we'll talk more about those in a moment. Right now, let's look at this concept of diploidy. Okay. We now know that if we took our chromosomes and we stretched them out, or looked at them, they're wiggly stuff most of the time. We stretch them out, we have sort of the stylized picture right here on the right. Okay, and on that we can have some genetic information, like gene A might control hair color, whether you have black hair, blonde hair, red hair, or whatever. Okay, where that gene is found on a chromosome and the gene itself is called a locus, right? The physical location of a gene. In fact, you'll do some labs where you locate the gene on chromosome 1, 2, 3, 4 of a fruit fly, and it's called the locus or location. All right. Now, as with fruit flies, they have 4 pairs of chromosomes, but they have thousands of genes. So there must be thousands of genes on a given chromosome. In fact, there are very often many different genes on a chromosome in different parts. So when you have more than one locus, the plural of that is loci, so the physical location of 2 or more genes, and they may not be on the same chromosome. You could have the loci. One could be on chromosome 4 and the other could be chromosome 12, okay? But you still say loci. They don't have to be physically linked together, but that's just an example. So locus we'll hear, all right? Okay, now what about this diploid stuff? Well, if you think about it, these organisms like us get one chromosome. We get 23 pairs in humans from our one parent, 23 pairs from the other parent. In fruit flies, we get 4 pairs or 4 chromosomes from one, 4 from the other. So we have 4 pairs or 8 chromosomes. All right. We call these chromosomes that we get from each parent homologous chromosomes, and homologs for short. Okay, we say that we're diploid. We're 2N, the N representing the haploid number. So for every chromosome we get from one parent, we get another one from the other parent. Okay, homologs have some things that common. One, the chromosomes, this chromosome from one parent to the other are the same length. They'll have the centromeres in the same place, and we've talked about what a centromere might be. And now they have the loci, and the loci are in the same place. If the locus for hair color is up here on this chromosome number 2 that you got from mom, the hair color locus is going to be up the same spot on the chromosome you got from dad, on the homolog that you got from dad. Okay, so we have gene A, it's located in some spot like this, and it's in the same plot. Okay, those are called alleles. The two pieces of genetic information that are found on these two homologs are called alleles of each other. That's one of those problems, right? We've seen where we call chromosomes different things. This thing is a chromosome at sometimes, one of these is a chromosome, whatever. Different time, different constructs. Genes the same way. These are both genes, okay? They're the same gene at the same locus, but now we're going to call them alleles so we can kind of keep them separate. Allele means, yes, it's a gene for the same thing, it's at the same locus, but they may not necessarily have exactly the same genetic information, right? It's the gene for hair color, but this allele may give you blonde hair and this one may give you red hair or something, okay? All right. Diploids have two alleles at every locus, all right? Basic idea. If those two pieces of genetic information are the same, whatever they are, this is black hair, you got black hair from dad, black hair from mom, you are a homozygote. Red hair from mom, red hair from dad, you're a homozygote. They're homozygous, okay? All right. If they differ, if you got a piece of genetic information from mom that told you to be blonde and a piece of genetic information from dad that said you should have black hair, then you're a heterozygote. So homozygous is the same, heterozygotes are different. Now, the heterozygotes are always genetically the interesting ones because are you going to be blonde or black-headed? Are you going to be blue-eyed or green-eyed? Are you going to be tall or short or wrinkled or smooth? Which one's going to win out becomes the really important concept, okay? So we now have homozygotes and heterozygotes. We have these concept of diploids, all right? Mendel answered that question that I just gave you. Which one of these wins? He said, well, one of them always wins, and we're going to call that the dominant one. And the one that loses, we're going to call the recessive, okay? Basically, when he said, when you have two unlike alleles, when you have two alleles that are same locus, different, slightly different genetic information, in other words, mutations of each other, which one is going to win? One will win out, okay? And that will be the dominant one, and the other one will be the recessive one. So he's talking about when you're in a heterozygous condition, the cell, basically, be anthropomorphic here, makes a decision about whether to make you blonde or whether to make you black-headed, right? We call that the mode of inheritance. This is your first mode of inheritance. It's all you need to work through many of the problems in those first chapters of the book. This is called dominant recessive. That's the mode of inheritance, or many times it's called simple Mendelian. Okay, you'll see it both ways. Both terms mean the same. One of those alleles is going to win over the other, absolutely. Green's going to win over yellow. Even if you got both pieces of genetic information, mom sent you down yellow, dad sent you down green, in the case of his stuff, green's going to win, or smooth's going to win over wrinkled. It's absolute. That's our mode of inheritance, okay? All right. Have to be careful. You go on the web or you look on other books or many places, they will say that Mendel had 4 basic principles. I don't think that these first 2 are really important enough to be principles. We don't have many laws or principles in biology like they do in physics or places. And principles are pretty important. They're things that really have to stand out and tell us this is a real piece of something that happens in nature and it's going to happen almost all the time. We're not all diploids, and not all organisms are diploids. And certainly dominant recessive, while it's a very, very common mode of inheritance, there are many, many, many modes of inheritance. In fact, we're going to spend the next couple of weeks working on other modes of inheritance, right? So those are ideas, they're important, but they're not principles. I'm going to say there's 2 principles, okay? And we're going to talk about the first one today. The first principle is what we saw going on in a monohybrid cross. It's what's known as the principle of segregation. And it says during the formation of gametes, these 2 alleles that you have randomly separate. You can't predict whether they're going into this egg or what. Okay, you can make an egg or you make a sperm or whatever you're making, a pollen or an ovum. You can't predict where the blonde one's going and this one. They're going to separate and it's going to be a random process. Okay. What he's talking about here is meiosis, because that's when we do the separations for production of gametes. He's talking about it being a random event, so we can't predict where it's going to go. There's no way to know where the yellow allele is going to end up in this particular sperm or not. And it occurs due to the separation of homologs, and we'll get back to that when we talk more about meiosis, okay, another one of your great favorite studies, okay? So he describes this, principle of segregation, Mendel's first principle. I will ask you over and over and over again what Mendel's first principle is. It will be on tests. It'll be all over the place. You should know exactly what it is and what it means, okay? All right, so let's go back to his monohybrid cross. We crossed a green with a yellow. We got all green. Not what we expected, but that's what he got. Now, I said, when I first set this cross up in the first lecture, I said, these are special parents. What I meant by that were they were what were called true breeding. We would call them homozygotes. True breeding in plants mean if you took this green and you crossed it with itself, which you could do, you would always get green. Or if you crossed it with one that had the same genotype, you'd always get green. You never see anything coming out. The yellows were the same way. If you would cross those with themselves, you'd always get it. That's pure breeding, and it means they're both homozygous. Okay. So if we were to put genotypes to that, we can use any letters we want, but we'll put big G, big G. They're homozygous green, and these guys are homozygous yellow. Okay. We could put a little Y there if you want to. I don't care, but just put two of the same alleles. So I'll just use little G's. Right. The little yellow thing here, the little dot indicates the gametes. Well, homozygotes can only make one type of gamete. They can make millions of them, pollen, but they're only going to have one genetic piece of information. That's the green allele, right? The yellow ones, if they're homozygous, can only make one type of gamete, and that's going to have the yellow piece of genetic information. We're calling that little g. Well, if you're pollinating those two together, the kids can only get a big G and a little g, right? That's all they can get. You're going to guarantee in the first filial generation that you're going to make all heterozygotes, right? So now what you're doing is you're crossing a het times a het, right? When you make the F2, classic F1 cross to go ahead and make the second filial generation. And when you do that, you've got a slightly different story, right? Because now each of these parents has two pieces of genetic information. They can't just put out big G all the time because they're heterozygous. So approximately half of their gametes are going to carry the green piece of genetic information, big G, and the other half is going to carry yellow or little g, right? And that's going to occur for both of them. So we're going to put all those together, okay? Now when we do that in the F2, we end up with this nice little 3-to-1 ratio down here, okay? Because why? Because we have this mode of inheritance playing a role. We have this dominance. Big G, green, is dominant. It doesn't matter what the other allele is. You get one big G, you're green, pure green, okay? To be yellow, you have to have two little G's. That's the only way that will work, okay? Now, it turns out that there are 6 crosses. That's all you got to know. 6 crosses for the entire section on transmission genetics. 6 crosses. If you'll learn these, if you'll know them, there's a printout sheet available on the web in our part of the modules. Print that thing out, lay it down, and anytime you're working a problem, have it next to you. And if you do, it'll be easy, okay? There's 6 crosses. That's all there are. You could cross a homozygote times a homozygote, the recessive homozygote times that. You can back cross, you can do these things. They all have different names, okay? But learn these and learn what they produce as far as genotypes, okay? So the first one is you cross a pure cross. This is to check to see if something is pure breeding. So taking that green one and crossing with it with self, okay? That's a pure dominant cross or pure big A. You could cross the other ones, the yellow ones with themselves. That's little a times little a, or little g, little g. We use G's. Earlier, that's a pure cross. You're not going to get anything but yellows or little g's or little a's in our case, okay? Then you start to get the interesting ones. Pure breeding, big A times pure breeding, little a, cross that. That's a parental cross. We expect all of our progeny to be hets. We cross a het times a het, which we just did. We see that we get this 1 to 2 to 1 genotypic ratio, which under Mendel, under simple Mendelian logic, turns into a 3 to 1 phenotypic ratio. And then another critical cross. We cross what's called a back cross, or just a recessive cross, where we take that het, that F1, and we cross it with little a, and we get a 1 to 1 ratio. Okay? That 1 to 1 ratio is the genotypes equal the phenotypes. We can tell what's going on. Critical cross. That cross will be a part of about half the genetics problems you ever work, okay? Between it and the other one, all right? You can also cross back to the dominant, but given that it's not going to show you much in this dominant recessive system because everybody's going to be the dominant, right? All right. So we have this thing called a test cross. Well, I said that was an important one when you had 2 little a's. We'll test an unknown against a recessive, homozygous recessive, and that's called a test cross. Almost every problem you do, somewhere in it has a test cross in it. It has some setup like that, so you can work with it. So a test cross And then these two parental crosses are the classic crosses that you need to know. And you need to know them instantly. People come into my office and they're working a problem and they've got big B little b times big B little b, and they're staring at it, trying to figure out what to do. They're doing a Punnett square or something and trying to figure out how many big B's and all that. And it's just instant. It has to be like that. You have to look at big B little b times big B little b and go, gives me a 1 to 2 to 1 genotypic, gives me a 3 to 1 phenotypic. It's got to. Once you can do that, you can move on. Until you can do that, don't even try to take the first exam or anything, because you're not going to be ready. You need to know this. Just learn this, okay? And I'm going to just really emphasize that over and over again. Okay. Now, something I want to point out. For now, we can learn this chart, genotypes and phenotypes. Notice that the phenotypes have turned red here. The reason for that is we're going to change modes of inheritance. We're going to talk about different modes of inheritance. The genotypes that you get from these 6 crosses will never change. If you cross a heterozygote, big A little a times little a, you will get a 1-to-1 expected ratio in the progeny of big A little a times to little a, right? Genotypically, that will always be there. That won't necessarily always give you a 1-to-1 phenotypic ratio, because the mode of inheritance may change on us, and that's all that's going to happen. We're always going to get that. But understand that what you're going to get genetically, we can just play a little bit with adding the modes of inheritance. So really, really important that you know that cross. Okay. All right. So Mendel's Big Six. Got all those. We've set up this cross. We've explained how Mendel got his. Now what we want to do is we're going to come back and we work a few problems. Okay. Just make sure that you see how you set a problem up. What's the simplest way to do it? And how do we make it work?